Funding for this program is provided by Annenberg CPD to advance excellent teaching.
Looking around at the multitude of life forms on Earth, it is often the differences that are most apparent.
For example, look at a face of a fly, and then look at your own face.
But the deeper you look, the fewer differences you see. Embryos of fish, or fowl, or human are remarkably similar in shape. In fact, the same genetic toolkit is used throughout their development.
To think that the same genes are controlling, the same processes are controlling the way a fly's eye develops, the way a fly's antennae develop, and the way your limbs develop, the way your eyes develop, the way your brain develops, it's utterly mind-boggling.
Picking through them one by one, mosaic tiles all look pretty much the same. And yet, in the right combinations, these small shards of color can become an infinite variety of complex art. Each tile is shaped and placed in a mosaic by an artist who creates the overall design and decides where each color and shape should go. But what if every tile had to find its own way, decide what color to become and how it should fit in with the tiles around it? This is exactly the task faced by billions of cells in developing plants and animals. Now, the latest techniques in molecular genetics allow scientists to better understand the generation of the whole from the perspective of the individual part and reveal the similarities that are shared by many different forms of life.
There are a number of tools that have really dramatically improved our ability to investigate, to compare the developmental mechanisms of different species.
John Postlewaite and his colleagues at the University of Oregon use zebrafish to study the evolution and function of genes.
A newly fertilized egg has all the DNA, all the genes, and as it develops, as it divides into thousands, millions of cells, each of those cells also has all of the genes, but they use only certain genes. And as they use only certain genes, their capacity to develop the number of different types of cells they can develop into decreases.
What are the genetic tools that chart the path a cell takes along its journey from embryo to adult? And what can be learned from stem cells, which even in adult form retain some ability to determine the direction of their final development? Thanks to great advances in genetic research, some of these questions have been answered. In most cases, before an artist begins cementing tiles of a mosaic into place, he creates a sketch or a pattern to guide the project. Living organisms also lay out the basic pattern for their development at an early stage, some of which involves an important component called the homeotic genes. Nipam Patel at the University of Chicago studies the role of homeotic genes in insects and crustaceans.
The normal function of a homeotic gene is to define the part of the body that should be in an appropriate place along its anterior-posterior axis.
Some homeotic genes, called Hox genes, contain a nucleotide sequence 180 base pairs long. This sequence is called the homeobox.
For one example, in an insect there are 3 thoracic segments. And of course it's very important that those segments be in that precise order. And these genes have to be expressed at the right time and place for the cells to know that that's the part of the animal they're supposed to make. So when a Hox gene is inappropriately expressed, you get a transformation of one part of the body into another part of the body. An example of a homeotic transformation is seen when the ultrabithorax gene, also called the UBX gene, is inappropriately expressed. An adult fly has its pair of wings on the 2nd thoracic segment. On the 3rd thoracic segment, instead of wings, it has little sort of gyroscope structures called halteres. If we remove the UBX gene expression in that third thoracic segment, what you get is really an amazing result. You get a fly with 2 pairs of wings, 4 wings. And what's happened is the cells essentially think they must be part of the second thoracic segment because they're not expressing UBX. And so they develop appropriately, but what's appropriate is now to make wings, and so you end up with extra pair of wings. Although our work is focused primarily on understanding the potential role of Hox genes in the evolution of insects and crustaceans, the work we do is still very relevant to understanding the normal function of these genes in all organisms, including vertebrates and including humans. By these increasingly sophisticated comparative studies in different organisms, it gives us actually more insight into making predictions about what these genes do in humans, how their expression is controlled, and what's the outcome of changing their patterns of expression.
Another feature of Hox genes is collinearity. Genes involved in the head formation are located toward one end of the chromosome, and those linked to the posterior are toward the other end.
One of the properties of homeotic gene expression in vertebrates is not only that they're collinear in terms of spatial expression, but that the Hox genes in vertebrates also show something called temporal collinearity. They also come on in a precise order, that the ones at the more anterior part of the animal come on before the ones in the more posterior part.
Each of the proteins produced from Hox genes contains a homeodomain, consisting of 60 amino acids encoded by the 180 nucleotide homeobox. This allows the protein to bind directly to DNA as a transcription factor, activating other genes, and thereby directing pattern formation in the developing embryo.
How do we know that Hox genes are important in regulating development from the anterior to posterior axis? We know that because when Hox gene expression is perturbed, then the embryo doesn't develop well.
In vertebrates, a retinoic acid gradient triggers Hox genes to be expressed in a particular order. If excess amounts of retinoic acid are present, genes requiring higher concentrations of retinoic acid will be expressed in inappropriate regions of the embryo. This will result in malformation of the embryo.
Now, one way you can get retinoic acid being present inappropriately is if a woman who's pregnant takes a substance that contains retinoic acid activity. One such substance is Accutane, a medicine commonly used to control acne. If a woman is pregnant, then that Accutane can cause the embryo to express its Hox genes inappropriately, and then the brain and the face of that embryo will be made inappropriately.
Researchers have confirmed that Hox genes are crucial to the embryonic development of many species.
So we can take zebrafish embryos and culture them in media that contains excess amount of retinoic acid. It causes the head and the brain of the embryo to be malformed. It causes Hox genes to be expressed where normally they wouldn't be expressed. It's utterly amazing to me that the similar genes can control development in such a wide variety of creatures.
As development of the embryo continues, a chain of events determines the fate of cells within each segment. As a result, cells receive the information they need to change shape and function and move into position to take on their adult roles. This is the process of cell-cell signaling.
Cell signaling involves one cell sending a molecular messenger to another cell. The other cell receives that signal that flips a genetic switch in the recipient cell, which then causes new proteins to be made, which then alters the activities of the recipient cell.
One example of this cascade of genetic switches and protein signals involves a gene that, when mutated, produces Drosophila larvae that seem rolled up and bristly.
Drosophila, the genes are often named for the phenotype that they create in the animal when the gene is mutated. When the hedgehog gene is mutated, you actually get a larvae which has hairs all over it and somewhat resembles a hedgehog. Now, often then, scientists will look for related genes in other organisms. So in the case of hedgehog, some of the scientists who were studying them called one of the related genes Sonic Hedgehog, which is sort of a play on words because it comes from the character Sonic the Hedgehog.
Hedgehog in flies and Sonic Hedgehog in vertebrates are genes that encode signaling proteins that are released, travel to a neighboring cell, and bind to its receptor, freeing an intracellular signaling molecule. That signal then activates genes that influence the development of the cell.
Hedgehog signaling is important for organizing the central nervous system in the dorsal to ventral aspect. The notochord sends out a hedgehog signal. It makes the hedgehog protein, which then diffuses from the notochord up to the spinal cord. Cells in the spinal cord then receive that hedgehog signal, and that tells them that they're supposed to be the ventral part of the spinal cord. Then the ventral part of the spinal cord makes motor neurons that controls our muscular activity.
Judith Eisen at the University of Oregon studies the development of neural cells in zebrafish.
So the history of zebrafish use really started here at the University of Oregon with George Streisinger, who was the first person to try to use zebrafish for genetic analysis of vertebrate development.
Zebrafish have many advantages over other organisms for use in embryonic analysis. They are raised easily in a laboratory setting. They breed readily all year round, and their embryos are clear and develop outside the mother.
Fertilization and the subsequent events occur in the water column, and so we can collect the eggs even before development starts, and we can watch every stage of embryogenesis as it unfolds. So one of the big questions that we're interested in my lab is how motor neurons form.
Researchers have discovered that with respect to the sonic hedgehog protein, the effect on cells depends on the doses they receive.
We know from work in several other laboratories that a signaling molecule called sonic hedgehog is particularly important during the formation of motor neurons. In fact, it seems to be necessary for their formation. Hedgehog proteins, and particularly sonic hedgehog protein, in other animals has been shown to act in a gradient fashion. What that means is different levels of sonic hedgehog protein cause formation of different types of cells. What we mostly look at are the genes that cells express and what those cells turn into. So we know that some of those genes directly respond to hedgehog, and that different levels of hedgehog are required to turn them on. That gives us a readout that the levels of hedgehog protein start in one particular place, and they move away from that place, and as they do, the concentration decreases, so that the way they interact in terms of turning on other genes changes as a function of the concentration.
This phenomenon is called a protein gradient. It is one example of how cell-to-cell signaling influences both the spatial arrangement and the function of cells in the developing embryo. But embryonic development, even within the uterus of mammals, is also affected by outside influences. Half a century ago, a surprising outbreak of birth defects among sheep led investigators along a trail that ultimately linked ancient Greek mythology, traditional shepherds, and the latest discoveries about environmental influences on the workings of the sonic hedgehog gene and protein.
From around the turn of the century up until the 1950s, there was always a low level of the birth of cyclopic lambs on sheep ranches in the Rocky Mountains.
Cyclopia is a developmental disorder that, in its extreme forms, involves the failure of the forebrain to cause the single, centrally located embryonic field to separate into two distinct left and right eye fields. This produces a cyclopic individual. John Incardona studies the effects of cyclopamine on chick embryos at the University of Washington.
In the 1950s, there were a couple of ranches, particularly in the Sawtooth Mountain regions of Idaho, that all of a sudden were having 25% of the lambs being born with this malformation. Couldn't really ignore it anymore. So that's when they got the U.S. Department of Agriculture involved to try to figure out what was causing the malformations. The first plants that they tested didn't have any effect, and they were actually helped out by one of the sheep herders that worked on the ranch, who was astute enough to notice that there was one particularly common plant that actually made the pregnant ewes vomit if they ate a lot of it.
That plant was Veratrum californicum, or the corn lily. The USDA scientists found that when they fed the corn lily plants to pregnant sheep, the sheep bore cyclopic lambs. The USDA's research methods could not determine exactly how cyclopamine disrupted development, but years later, more advanced genetic techniques would.
So they were then able to purify large quantities of cyclopamine in pure form, and that later, much later, decades later, allowed us to do much more precise mechanistic studies to determine how cyclopamine caused these horrendous malformations. And this is where the genetics really helped out. Now, by applying cyclopamine to chick embryos, we can induce the same malformations, but now we have the molecular tools, such as antibodies, for specific cell types in the nervous system, to go in and ask, is the hedgehog signaling pathway disrupted by cyclopamine?
Studying the thread that connects the sonic hedgehog gene to Drosophila may lead to new treatments for other diseases, including cancers. Intentionally disrupting developmental signals in the sonic hedgehog pathway might interfere with tumor growth.
Because cyclopamine is actually a receptor antagonist, it could potentially be used to shut down the pathway in tumors in which it's activated, and therefore be used as a treatment to stop growth of the tumor.
Patterning and segmentation genes and the hedgehog gene pathway are common genes of development shared by many species. They comprise a sort of genetic toolkit.
Because different organisms look so different, a fly looks so different from a human, we used to think that their developmental regulatory mechanisms must be very, very different. However, we've discovered that the genetic toolkit used by a fly and a human and most other animals is very, very similar.
There's sort of basic genes that are used anytime you want to build any sort of structure or pattern any sort of structure in a developing animal.
Just like if you're creating a mosaic, assembling a car, or building a house, there's a similar toolkit used even though each project seems very different from the other. The genetic toolkit, then, is a core set of genes that builds upon fundamental steps of development that we now know are essentially the same in a fly, a fish, or a person. Much has been revealed about the journey a single cell takes to a fully formed organism. And with this knowledge, there may be opportunities to control the mechanisms of development for therapeutic purposes. Marcus Grompe conducts stem cell research at Oregon Health and Science University.
A stem cell is a cell in the body that is responsible for renewing other tissues. So it itself is not a differentiated functioning cell, but it is a cell that's sort of the reservoir for other cells that are needed in the body. Embryos at conception are totipotent, and as the number of cells in the embryo increases, their developmental potential becomes further and further restricted.
A fertilized egg is an example of a totipotent cell. It can produce any type of cell used within an organism. But as cells mature, they go through a process called differentiation. The inner mast cells of a blastocyst become more differentiated and are called pluripotent, meaning they can produce most but not all types of cells. Eventually, the cells in an adult organism become multipotent and can produce only a defined set of mature cells.
So basically, you start with a totipotent cell, and as the number of cells increases, their developmental potential becomes increasingly restricted. So that in the adult organism, there are lots and lots of cells that can be only one thing, and very few cells that have multiple potential.
While all these cells are called stem cells, there are critical distinctions between stem cells that come from an embryo and those derived from adults. Distinctions that are both technical and ethical.
Embryonic stem cells and adult stem cells can be both used for research. There are several advantages and disadvantages to both. The most important advantage of embryonic stem cells is that they can be easily grown to large numbers. It takes a very short period of time to grow a lot of them, which makes it easy to study. Also, the fact that they can develop into multiple tissues makes it possible to study those processes. The disadvantage of adult stem cells in comparison is that they grow much more slowly and at lower density, so that it takes a long time to make lots of adult stem cells. However, the advantage of using the multipotent adult progenitor cells is that they could potentially teach you about developmental processes in the adult, which could be very different from embryonic stem cells.
There are also differences in the clinical application of embryonic and adult stem cells.
The most important one is that embryonic stem cells are derived from donor fetuses that are immunologically mismatched to the person you'd want to be treating. So you will have the same issues as you currently have with organ transplantation or with blood transfusions, where you might have to actually use immunosuppression to be able to use those cells. With the adult stem cells, it should be possible to derive those cells from the patient, him or herself, that needs the cells, so that you, at least in theory, may be able to get away completely from using immune suppression.
Some researchers are optimistic that if adult stem cells from a patient could be genetically corrected in vitro and then cultivated to produce the type of mature cells needed for treatment, the immunorejection problem might be avoided. But another challenge is that adult stem cells can take longer to cultivate, and they typically produce only closely related types of mature cells. However, in some experiments, that natural limitation appears to have been overcome.
There are now the multipotent bone marrow cells, which have been shown, at least in tissue culture, to differentiate into liver cells, neurons, muscle cells. Virtually every cell type that has been seen in embryonic stem cell differentiation has also been seen with these multipotent adult progenitor cells.
Despite the obstacles, stem cell research is pressing ahead because the potential benefits could be enormous. People with Parkinson's might receive new dopamine-producing brain cells. An infusion of stem cells primed to become heart muscle could repair the damage of a heart attack. Or a patient might receive new insulin-producing pancreatic cells.
The hope for stem cells in the area of diabetes lies primarily in the area of type 1 diabetes, where the insulin-producing beta cells of the pancreas have been lost. It's been actually shown that it is possible to take undifferentiated embryonic stem cells from the mouse and turn them into insulin-producing cells. And most recently, actually, mice with the equivalent of type 1 diabetes have been treated and successfully with embryonic stem cell-derived beta cells.
Many researchers are now working toward the next steps.
The current main interest of the field is how to get multipotent stem cells to become differentiated cells. That's certainly very important, but it only gets us halfway to therapy. The second really, really important component to using cells for therapy is to actually get them into the patient. Many people are under the impression that just transplanting stem cells is somehow miraculously going to result in a cure. What I'm convinced of is that we need to learn how to get these stem cells to grow better than the endogenous cells upon transplantation. It's not the cell. I'd like to say in my lab, it's not the cell, it's how you get it to grow.
Science and technology have identified which therapies may someday come from harvesting embryonic and adult stem cells. But who decides what research is acceptable or appropriate, and where those lines are drawn?
People under the impression that embryonic stem cells are little people being grown in the tissue culture dish in the lab, and that's really, these are not embryos. They're embryo-derived, so in the process of generating embryonic stem cells, an embryo is destroyed, but it's not like the same thing as cloning or actually having the equivalent to a human conceptus in the lab.
Government regulations typically define what is legal or not, but even those lines can fluctuate because as the field of genetic research advances, its ethical boundaries are tested each step of the way. What one group considers moral may not be by another. Those same developments and discoveries that rapidly push the field forward also fuel the ardent protests and ethical disputes.
And the ethical debate boils down to when does human life start? Is it at conception with the potential to develop into a full human being, or at some later stage? And obviously, people do not agree on that particular point.
Deciding what therapies are appropriate, what research is permissible, and by what means sparks discussion by both the scientific and non-scientific communities. None of these questions have an easy answer, however, and most likely will be debated as long as the desire and need for exploration continue. But dramatic advances in the research of genetics have allowed scientists to take enormous steps toward understanding the common denominators of species. It has enabled them to compare the development of model organisms and better understand those same developmental processes in humans.
It's truly amazing how similar the action of genes are from very, very different species. You know, 20 years ago, 30 years ago, we wouldn't have realized that.
Understanding cell differentiation may bring to light new therapies for diseases. And the discovery of a genetic toolkit shared by many species further galvanizes the theory of development that links all living organisms.
One of the fascinating things about evolution and the evolution of development is the fact that these organisms that are evolutionarily much older are using these basic toolkits that were established in organisms hundreds of millions of years before even vertebrates evolved. So the same genes are used over and over again throughout evolution to solve similar types of problems for the developing organism.
Finally, the question of ethics will always be central in deciding what research should be pursued, as will the desire to more fully understand the development of the living organism, thus provoking impassioned debates between conflicting communities. But the recently discovered similarities shared by so many species are undeniable, and it seems evident now that all life on Earth is more closely connected than ever realized.
Funding for this program is provided by Annenberg CPB to advance excellent teaching. For information about this and other Annenberg CPB programs, call 1-800-LEARNER and visit us at www.learner.org.